I hope you all had a nice break. Our next session brings together voices from the biotech sector, companies working on some of the most promising and potential life-changing therapies for inherited retinal conditions. In this panel session, you'll hear from Dr. Jayashree Sahni, Chief Medical Officer at AAVantgarde, Julia Carpenter-Conlin, Head of Patient Advocacy at Alkeus Pharmaceuticals, Dr. Dan Chung, Chief Medical Officer at Beacon Therapeutics, Dr. Matthew Albert, Vice President of Discovery Biology and Early Clinical Development at Octant, and Dr. Andy Bolan, Patient Advocacy Director at Sepul Bio. Each will spend 5 minutes updating you on their activities. Some of them will also be available this afternoon to take questions. Let's welcome them to the stage.
Good morning, everyone. It has been a really interesting morning, and I'm very grateful for the opportunity to be here. My name is Jayashree Sahni. I'm the Chief Medical Officer at AAVantgarde. But in my previous life, I was a retina specialist, an IRD specialist, and I also trained at Moorfields under Tony Moore. And it's a great honor to be representing AAVantgarde here. We are a clinical stage gene therapy company with our first assets in rare diseases with high unmet need. So this is our pipeline. As you can see, our first indications are 2 diseases, which you're probably familiar with, and I'll tell you more about it very shortly. And we also have an undisclosed pipeline in other rare diseases, and our mission is to transform the life by innovative gene therapy. So as I mentioned, we are addressing 2 inherited retinal diseases with a very high unmet need. The first of it is Usher syndrome. Many of you may know about it. A syndrome has a triad, so it has the patients gradually lose their vision. In addition to that, they also have hearing loss and some balance issues. Usher 1B, which is the disease that we are focusing on, the children are born profoundly deaf. They may get a cochlear implant and they could effectively hear later. However, they do lose vision, and at the moment there is no treatment for that. One of the challenges with the gene that is associated with Usher 1B is the gene is very big. You heard from Professor Michaelides about RPE65. There we are using a virus to transport the gene into the patient's eye. So here, because the gene is bigger and cannot fit into one virus, we have to use 2 viruses. We split the gene, and we use a technology which our founder, Alberto Auricchio, has done. So the gene or the DNA is split somewhere in the middle. He has a unique splitting point, and then he can take those 2 and he can put it into the virus. And then when the virus infects the cells, so myo7A is expressed in the photoreceptors as well as the RPE cells. And when the gene is expressed in the RPE cells and the photoreceptors, in his animal models, it did show a restoration of the protein function. With that information, we were able to move into phase 1 trials. Our phase 1 trial is in Europe as well as in UK. And we have, as of now, dosed 11 patients. We are very pleased to say that we have gone through the safety rigors. We have an independent data monitoring committee, and we are now on the high dose. And we have also presented the data at several conferences, and we are keeping our fingers crossed that we will be able to bring a therapy to patients. We are looking at endpoints. As you know, visual function, Professor Michaelides again mentioned, there is anatomical as well as functional benefits, and we are looking at different parameters, and we are very grateful to the sites as well as our patients who go through a lot of tests so that we can find a signal. And we are seeing some early signals, but it's very early, so we would like to wait a bit longer to be absolutely certain. Once this study goes ahead, and if we are successful in a year or 2, we will be able to advance further. The second indication, which you know about, is Stargardt's. So unlike myo7A Usher retinitis pigmentosa, where the sight goes from outside in and the patient is left with a tunnel vision, here the central vision is lost. Again, it sets in quite early, depending on your mutation-specific variability, etc. and they go blind from the center out. And that is caused by another large gene. So we at AAVantgarde have a proprietary technology to deliver large genes. And here we are delivering the ABCA4 gene. Here we are using another of our methodology to deliver it as a protein, and it is very fast and very efficient. And we have demonstrated it in multiple animal models. And at the moment, we are conducting natural history studies globally in UK, in Europe, as well as in US. And we are on the threshold of starting our interventional trial, which should be any day now. So I just wanted to tell you that we are hoping that we will be able to bring good news in the future to this community. Thank you for all your help.
Hello, everyone. My name is Julia Carpenter-Conlon, and I lead patient advocacy at Alkeus Pharmaceuticals. I am stepping in for our Chief Medical Officer today, who at the last minute was unable to make it. I do want to say thank you so much to the Retina UK team, Kate and Matt and James, and everybody has just been really incredible, and I enjoyed the day yesterday greatly. So, there we go. Oh, I should have told you. Hold on one second. I just want to also say that Jack, on the right, I want to describe this slide, is one of the patients that's in my patient advisory board, and that is his guide dog, Adonis, and he has really helped us in developing all of our patient-facing materials. So, Alkeus, and on this slide, there's also a picture of a young boy. He does not have Stargardt disease, and so that's really just a stock image. But Alkeus So Alkeus is a clinical stage biopharma company located in Cambridge, Massachusetts. We're just outside the Harvard University campus. We are dedicated to preserving the sight of people impacted by retinal diseases, and that purpose is really at the heart of what we do. So our initial focus is in Stargardt disease. And our investigational therapy, gilduretinol, is a once-daily oral pill designed to reduce toxic dimer formation without negatively impacting the visual cycle. And we have the following designations in the US and the EU. We have breakthrough therapy designation in the United States, orphan drug designation, rare pediatric designation, and orphan medical product designation in the EU. And a little bit about the company, and about Stargardt. So on the slide, on the left, we talk about Stargardt as a rare disease with high unmet need. The estimated incidence in the United States is about 1 in 8,000 to 10,000, and there are no treatment options today. Gilduretinol is our investigational therapy. It's a new molecular entity. It's an oral, once-daily investigational product, and it's designed to reduce toxic dimerization formation without negatively impacting the visual cycle. And it's designed to systematically metabolize, similar to vitamin A. So Stargardt is the most common form of juvenile macular dystrophy. And for a rare disease, it's fairly prevalent. And we know that Stargardt is a genetic condition, and what is important to note for our program is in the second box. And on that slide, it's the specific focus of our therapeutic program is in the ABCA4 gene. So we're focused on, let's see, hold on. The resulting accumulation of vitamin A dimers in the retina and their related toxicity for the retina is at the heart of our primary mechanism of disease. So as I mentioned earlier, our therapeutic approach focuses on the role of vitamin A dimers in the progression of disease. And vitamin A plays an important role in the retina for healthy vision. In Stargardt disease, pathogenic variants of the ABCA4 gene lead to an impaired transport of vitamin A in the retina. And as a result, vitamin A accumulates and forms dimers, or clumps of vitamin A, and those dimers create toxicity for the cells of the retina. So in this slide It is about gildaretinol, and gildaretinol is our investigational therapy. And as I said, it was designed specifically to address that issue. And as a new molecular entity delivered orally, it is recognized and metabolized by the body as if it were natural vitamin A. But in the retina, it clumps or dimerizes much less. And importantly, and for clarity, it is not a visual cycle modulator. It is designed to reduce dimerization, letting the natural biology do the work. So we are running a comprehensive clinical program which covers all stages of Stargardt disease, as well as geographic atrophy secondary to age-related macular degeneration. We have 4 clinical trials treating advanced disease, intermediate disease, and early disease. And we also have an open label extension study called the TEASE 2 trial. We also had a study in dry AMD looking at geographic atrophy. So part of the reason that I am here in the UK and I've been engaging with patients in the UK and in the EU is that we are planning for an international phase 3 study for gildaretinol in Stargardt disease. And one of the exciting things that I've had the opportunity to do is work with the Retina UK team and Stargardt's Connected and identifying people living with Stargardt disease who can help me in getting feedback on our protocol as part of our commitment to patient-focused drug development. So I really look forward to talking to folks later in the Meet the Speakers session, and thank you so much.
Well, good morning. It's great to be with you. Thank Retina UK for the kind opportunity to be here. My name is Dan Chung. I am the CMO of Beacon Therapeutics. I am a 62-year-old Korean-American man in a dark blue suit with rapidly graying hair and more pounds than my cardiologist would like. So on that note, I've been in the retinal gene therapy space for about 30 years. I was part of the team at University of Pennsylvania, Children's Hospital Philadelphia, and Spark Therapeutics that brought the first gene therapy known as Luxturna. And unfortunately, right now it is still the only. But there, as you heard earlier, there are many good folks working on a lot of different therapies that hopefully will get to the finish line very shortly. With that note, Beacon Therapeutics, we are a late-stage clinical gene therapy company only in the eye. We are working on an X-linked form of retinitis pigmentosa, looking at the gene RPGR. We also will have a program in dry AMD, hopefully in the next year. And right now we're also considering a cone rod dystrophy program as well, but that is still very much in the early stages. So for many of you, I don't have to talk too much about retinitis pigmentosa, but what we are targeting is the X-linked form, which is about 10 to 20% of all RPs. And RPGR accounts for about 70% of those X-linked forms. And as you know, this is a progressive disease that unfortunately degrades the vision starting from the periphery. But RPGR also affects the central cone cells as well. So obviously a disease that we all want to try and treat. So for us, we're using gene therapy. We're looking at a full-length gene with the whole gene encoded. It's codon optimized. And this is also into an AAV vector put into the subretinal space. So right now, there are many different trials. This was an asset that a company called AGTC had originally worked on. And then a British venture capital group actually bought that when, unfortunately, they had run out of money, called Syncona. And they began Beacon Therapeutics, where we took the vector and optimized it in many ways. So the first couple of trials was really about looking at different doses to see which is the best. In the second trial, we looked at something called microperimetry, which talks about the sensitivity of the retina, and can we see some changes after gene therapy, which we did. And then the last phase 2 trial that has been completed looked at something called low luminance visual acuity, and that was the primary endpoint of our primary regulatory pivotal trial known as VISTA, which just completed their phase 3 enrollment and endorsing this past July. We hope to have results of that in the second half of 26. And obviously, our phase 2s are relatively small and the results are promising, so you can't always guarantee that's what you're gonna see in your final pivotal trial of a phase 3, but we are hopeful. And as previous speakers say, I used to always say cautiously optimistic, and now maybe I'll now say optimistic and drop the cautiously. So we'll see how that goes. But we are very much looking forward to the results in the next year. For that, many things we have seen in the early trials, and that is that there's good safety. The subretinal injection is well tolerated. We have a very stringent steroid procedure to make sure inflammation goes down and is not a problem in our trials. So we are very hopeful. We are optimistic that in this next year we will have an answer to our trials, and we can hopefully, if I can be a part of another approval, then I will definitely be very humbled by that. So with that, this is a image up here, is simply a trial of the Dawn, which was the second phase 2 study. 15 patients were done. There was a low and a high dose. And again, as I mentioned, we looked at low luminance visual acuity, which the FDA has already said they would take as a primary endpoint. We talked to them about our trial design for VISTA, which was the phase 3 trial. They also said that was very acceptable. So we have gone through many of the regulatory hurdles, but we have met many of those challenges, and we believe that there is a good path forward. This grid simply is, there's a couple graphs on this slide, and simply showing that the improvements in low luminance visual acuity, most patients have gained about 10 letters and some 15 letters. So we are just watching these, but again, these are small numbers, and in our pivotal trial, it was 84 patients. So we are hopeful on that. So this is just another schematic that you see. It's a chart here with 3 different lines, and it just shows that there are 3 different groups in our VISTA trial, which is our final trial, 25 patients in each. We actually enrolled 84 total. So again, as I said, we are done with that, and we are looking forward to the results very soon. So with that, I'm just going to end and simply say that we'll keep our fingers crossed, and we'll hope for the best, and hopefully we'll be back next year to give you some good news.
Oh, Octant, sorry.
I was getting very excited that you were going to give my talk for me. Hi, everyone. My name is Matthew Albert, and I'm an American. Really excited to be here. On behalf of Octant Biosciences, we're a San Francisco biotech company. I'm 5'10", wearing a blue checkered shirt. I've got brown hair, but it's thinning, pulled back in a ponytail, and I've got blue readers, and it's very confusing because I need them to read the text, but also want to engage with you as the audience. As I was saying, it's a real privilege to be here, and I'm really grateful to Retina UK for including Biotech Voices as part of this program. Over the past day and a half, I've met so many inspiring people and been really impressed by the work of the Retina UK over the past year as we've gotten to know them. For the next few minutes, I just want to share some of my thoughts about how human genetics is shaping new treatments for inherited retinal disease, how the Retina UK and everyone here today is so important in that process, and how participation in clinical trials is really what is key for turning genetic discoveries into real treatments. I want to begin by highlighting first how closely we are all connected. And I'm speaking of human genetics, really at the level of DNA. We are each 99.9% identical at the level of the letters in our DNA to any other person in this room, which is really quite remarkable. If you think about the small differences, and this is the tiny 0.1%, a fraction of the genetic code, that's what's shaping our individuality, our health, and also our risks for disease. In my own family, human genetics has played a very big role. My grandmother, my mother, my sister all faced cancer linked to a single gene. And that experience has really taught me how powerful genetics can be in shaping health and defining family stories. For this community, inherited retinal disease, genetics has absolutely been central. We heard that from many of the speakers over the past 2 days. And speaking to the complexity, there is now over 300 different genes that are associated with IRDs. And even within a single gene, I'm going to be talking about rhodopsin, different mutations can disrupt the retina in very different ways. It's why programs like Unlock Genetics, which Kate was talking about a little bit yesterday, a key initiative for Retina UK, is so important. They help families navigate testing, support people through the complex results, and really prepare patients to take part in clinical trials, such as the opportunities that I'm going to be talking about for Octant. And it's really the understanding of human genetics that's what's guiding our work. Our first clinical program is focused on rhodopsin-associated retinitis pigmentosa. This is the most common form of dominant RP. And our clinical candidate, which we call OCT-980, is designed as an oral therapy, a simple pill taken once a day that can help people with this condition. And in the lab and in early animal testing, what we've discovered is that OCT-980 reaches the retina through the bloodstream. It helps rhodopsin fold and correctly function, and it restores low light vision or night vision, and it improves rod cell survival. We believe 980 could potentially help about half of people with rhodopsin mutations, and if successful, it would be the first oral therapy that doesn't simply treat symptoms, but really gets to the root cause of disease and improves sensitivity to light, which is quite remarkable. So how does small molecules work? And I bring it up because it is a rather new thing for ophthalmology, as we've discovered. Unlike gene therapy, which requires surgery, we're talking about pills that are taken by mouth. The molecules are absorbed by the gut, and it travels through the bloodstream to the back of the eye. And it crosses what's called the blood-retinal barrier to get into the rod cells that are mediating the effects on rhodopsin. So to go one step further and sort of explain it at a molecular level, you can really think of the rhodopsin protein like a paper airplane. And if you come by my booth, you can pick up a sticker and you can see our octopus, which is our mascot, folding the paper airplane. If every fold is correct, you get a plane that flies. But if you have one wrong fold, then it doesn't. And similarly, a mutation in the rhodopsin protein causes the protein to misfold, and it doesn't reach the right place in the retina, and it can't sense light. What a corrector therapy does is it corrects the folding, helps it out of the machinery placed in the cell, unclogs what is a back jam in the cell that is causing cell stress and ultimately rod cell death, and it gets it to the right place, which relaxes that stress and allows the protein to start doing its job again. We've made a lot of progress, and we're really excited that we have a clinical candidate. We're just at the brink of submitting to regulatory authorities. The plan is to lead our first clinical studies in the UK with the first treatments by the end of the year. And the reason that we're here, many of us, is that really the next step requires deep collaboration with patients and families with retinitis pigmentosa. Genetic testing, registries, clinical trials, this is what converts the scientific ideas and the work that we've been doing over the past 4 years into real medicines. You enable our discoveries, and over the past year, it's really been a privilege getting to know folks within the community, working with the Retina UK, and we look forward to a partnership to be able to bring OCT-980 to patients. I truly believe that we're at the brink of a really new era where we're beginning to see genetics guide drug discovery and medicines that are designed to match the root cause of disease. Retinal disease may diminish light, but through the shared stories, genetic testings, clinical trials, that light can be recovered. And I believe that we can look towards a future where inherited retinal disease can be managed and ultimately overcome. Thank you, and I look forward to meeting you this afternoon with the speakers.
Right, my turn properly this time, so apologies, Matt. That was super elegant. Good afternoon, everyone. My name is Andy Bolan. I'm the Patient Advocacy Director for Sepul Bio, which is an innovative business unit of a company, Laboratoire Théa. that purchased 2 late-stage assets from a company called ProQR Therapeutics about 18 months ago. And some of us at the team in ProQR moved over to help set up the clinical programs and get them off the ground as quickly as possible. A huge thank you to the Retina UK team for inviting us to speak, and also the interpreters, because I'm going to use some very long language now, which I really hope and wish you best of luck. So we have 2 programs, Sepofarsen for CEP290 or CEP290, mediated Leber's congenital amaurosis type 10, and Ultevursen for USH2A or retinitis pigmentosa due to mutations in the variant exon 13 of the USH2A gene. And Sepofarsen is an RNA medicine, which means that it isn't a permanent DNA fix. We are looking at about 6 monthly dosings, and it is an antisense oligonucleotide. Or an AON, which makes it slightly easier to sign. It's delivered by intravitreal injection, or IVT, which is injected into the vitreous or the jelly-like area of the eye. And as I said, we're looking at about 6 months dosing for that. And it has all of the regulatory fast-track elements to it, which is really important for trying to bring this late-stage phase 3 molecule, hopefully, over the line. Changing track to Ultevursen, which is in phase 2, so the phase before the last phase of clinical development. And we've done a phase 1/2, which really showed some really encouraging signs. This is also administered through IVT and is looking to alleviate or stop in its tracks the degeneration of the retina due to variants in the exon 13 of the USH2A gene. And LUNA, our trial, has kicked off, and we have some fantastic sites in London, in Moorfields, in Oxford, and in Edinburgh, which is opening very soon. So where are we as a group? Well, it's very, very lucky that the team came over from ProQR because we managed to hit the ground running. It normally takes a couple of years for these trials to get off the ground, but LUNA is active, and we are actually looking at dosing our first individuals in the phase 3 Hyperion study in the next couple of weeks, which is really, really exciting. We wouldn't be able to do this with loads of help from the community. So Sepofarsen actually had a readout which didn't meet its primary endpoint, and in the industry, that can be a real death knell for a molecule. But I was part of the team that actually heard all of the community, the families who were part of the trial, who said, please keep us on the medicine. That was enough for a company like Laboratoire Théa to pick up the medicine from ProQR and carry on the development. And we're in that phase 3, that final area of development. So if you're really, as I draw this talk to a close, I really want to say a huge thank you to Retina UK, who have been there throughout the whole journey of these 2 molecules. They really have helped us with individuals to give us their lived experience, and I was here for the panel yesterday, and I cannot tell you how important it is. I left the room to take a call with colleagues, and I literally verbatim told some of the stories that we had heard, and it makes a massive impact for motivating, but also for learning why we're doing these products and these programs in the first place. So all of the individuals in this room, you have a story to tell, and it is super important for us as the industry to hear them because it makes our programs stronger. I also want to give a shout out to the potential of the patient registry. I know Matt and Kate have been working on it, and we work very closely, or I certainly do, with advocacy groups around the world. And one of the key elements of having a centralized registry is the power of you and your data. Now, having that centralized is an amazing tool, and we've already partnered with organizations in the US, and I'm going to do a partnership in Canada in the next couple of weeks as well. So that's an amazing piece of work with the potential to put the UK really at the heart of future research. But the power of research is only that of the people who are in it, and you of the community, I've always said, is the engine of research. Without people coming and joining our trials, they don't happen. They don't progress. And so actually, I always use the car analogy, you are the engine. We can build everything around you. We can do all the regulatory submissions. We can do all of the paperwork that's needed. But if people aren't willing to come into the clinics and take our medicines, they don't move forward. So with that, I'll say a huge thank you. I'm really looking forward to speaking to some of you later this afternoon. If you've got any questions, I should clear up, I'm not a doctor. So any compliance colleagues watching this online, I'm not a doctor, so I can't give medical advice, but I am an advocacy colleague for you to come and ask any questions and point you in the right directions. So thank you very much.